Every technology is a double-edged sword, and so it is with the technology of cloning. Along with the promise of medical breakthroughs and health benefits, come the prospect of ethical and moral dilemmas that may plague humanity for generations to come. Now the question is it really worth all the trouble? Or are we only opening the proverbial Pandora's box? The following will help you understand and decide on which side of this contentious issue you will stand. But before we go into details, let us first know what cloning is. Cloning is simply the creation of an organism that is an exact genetic copy of another. Basically, there are three different types of cloning. The first one is the embryo cloning, or the so-called artificial twinning. This process stimulates the mechanisms by which twins naturally develop. Nature itself is the greatest cloning agent. In about 1 of every 75 human conceptions, the fertilized ovum splits for some unknown reason and produces monozygotic twins, or what we usually call as identical twins. In cloning, this same operation is done intentionally in a laboratory. It involves removing one or more cells from an embryo and encouraging the cell to develop into a separate embryo. This diagram will show you on how embryo cloning is done in cow species. After the cloning process, the baby cows have the same genetic makeup from the donor organism. This type of cloning has been successfully carried out for years on many species of animals, and there were also some very limited experimentation that has been done on human embryos. The second type of cloning is the somatic cell nuclear transfer. This process uses a different approach than artificial embryo twinning, but it produces the same result. This was the method used to create Dolly the sheep. Ian Wilmot and a group of Scottish scientists dared to undergo the complicated process of cloning experiments. They started from isolating a somatic cell from an adult female sheep. Next, they transferred the nucleus from that cell to an egg cell from which the nucleus has been removed. After a couple of chemical tweaks, the egg cell with its new nucleus was behaving just like a freshly fertilized zygote. It developed into an embryo which was implanted into a surrogate mother and carried to term. The lamb Dolly was an exact genetic replica of the adult female sheep that donated the somatic cell nucleus to the egg. So on July 5, 1997, the most famous sheep in modern history was born. She was the first ever mammal to be cloned from an adult somatic cell. The third type of cloning is therapeutic cloning. This focuses on the production of human embryos for use in research. Its goal is not to create cloned human beings, but rather to harvest stem cells that can be used to study human development and to treat disease. So this is where stem cell technology comes in. What are stem cells? These are a class of undifferentiated cells that are able to differentiate into specialized cell types. Stem cells are important to biomedical researchers because they can be used to generate virtually any type of specialized cells in the human body. Stem cells are extracted from the egg after it has divided for 5 days, which is usually called as blastocyst. Many researchers hope that one day stem cells can be used to serve as replacement cells to treat heart disease, Alzheimer's, cancer, and other diseases. We might not be aware of it, but cloning technology may not only be a future lifesaver to human beings, but also to our biodiversity. First, cloning vital organs of human body can be used as backup in case of an organ failure. When a crucial body organ such as a kidney or heart fails to perform its normal functions, it can be replaced with a cloned organ substitute. This technique would be vastly superior to relying on organ transplants from other people. The supply would be unlimited, so there would be no waiting list. The tissue or organ would even have the sick person's original DNA, so the patient would not have to take immunosuppressant drugs for the rest of their life, as is now required after transplants. There would not be any danger of organ rejection. Second, cloning can also provide a viable solution to infertility in human beings. It can help the infertile individuals in producing children. Cloned embryos can be implanted into women's bodies to produce babies. Third, cloning can make it possible to reproduce a certain desirable trait in human beings through the cloned embryo. Not only that, cloning technologies may help to understand the composition of genes and their effect on human traits and behavior in a more comprehensive and elaborate manner. Fourth, cloning can make it possible to alter genetic constituents in cloned humans so as to simplify their analysis of genes. Because of this, a wide range of genetic diseases can be averted through cloning. Lastly, genetic alterations of plants and animals can also be enabled by cloning. It can also help to replicate animals that can be used for research purposes by scientists, and it can even help in saving endangered species. On January 8, 2001, scientists at Advanced Cell Technology Incorporated announced the birth of the first cloned endangered animal, a baby wild ox from India and Southeast Asia named Noah. Although Noah died of an infection unrelated to the procedure, the experiment demonstrated that it is possible to save endangered species through cloning. In 2002, geneticists at the Australian Museum announced that they had replicated the DNA of a Tasmanian tiger using a polymerase chain reaction. This thylacine or Tasmanian tiger is also an extinct organism. However, on February 15, 2005, the museum announced that it was stopping the project because the specimen's DNA had been too badly degraded by the ethanol preservative. On May 15, 2005, it was announced that the thylacine project would be revived with new participation from researchers in New South Wales and Victoria. In 2003, a banteng was successfully cloned, followed by 3 African wildcats from a thawed frozen embryo. These successes provided hope that similar techniques using surrogate mothers of another species might be used to clone extinct species. Anticipating this possibility, tissue samples from the last bucardo were frozen in liquid nitrogen immediately after it died in 2000. Researchers are also considering cloning endangered species such as the giant panda, ocelot, and the cheetah. The frozen zoo at the San Diego Zoo now stores frozen tissue from the world's rarest and most endangered species. As cloning technology develops, opposition from individuals, political groups, and religious authorities are also building up and point out several disadvantages. First, since cloning creates identical genes, and it is a process of replicating a complete genetic constitution, it can significantly hamper the much-needed DNA diversity in human beings. The lessening of genes diversity will weaken our adaptation ability. Similarly, cloning will also severely affect diversity in plants and animals. A cloned species may not know how to react to viruses and other destructive agents, as scientists cannot predict such potential developments. Second, by allowing man to interfere with genetics in human beings, cloning raises a concerning probability of deliberate reproduction of undesirable traits in human beings. Cloning of body organs opens the possibility of malpractice in medical fraternity. Third, technical and economic barriers need a consideration in cloning human organs for transplant. Cloned organs may not be cost-effective for a good part of human society. The benefits of cloning techniques reaching the common man remain a big question mark. Lastly, on moral and ethical front as well, cloning devalues mankind and seriously undermines the value of human life. So, is cloning bad or good? Well, it's up for you to decide and make a stand.